So first I want to thank the organizers for giving me this opportunity to present our results. And in my lab, I'm interested in the genetics underlying variability in skeletal muscles. And variability is quite extensive in human beings. If we look at the total body mass, the differences between individuals can range by more than twofold. And if we look all the way down to the muscle fibers, so the numbers of fibers in the soleus, in vastus lateralis or biceps brachii muscles, they can also differ by more than 2 times between individuals of the same age and the same sex. The same can be said about the proportion of oxidative fibers in the muscles. So all these obviously variables are important for athletic performance, and also they would affect the ability to adapt to training. So would help understanding of the mechanisms underlying this variability, and genes are playing an important role there, would help us to understand the also phenomenon of responders and non-responders. In my lab, we're using a mouse model to study these genetic effects. And it's a useful model because it allows us to control the environmental conditions quite well, much better than it can be achieved in human studies. It also allows us to do some invasive and accurate measurements of relevant phenotypes. And also we can use the animals of a very similar age, so we can reduce the effects of environmental factors, and then it becomes easier to tease out the effects of the genes. And in this presentation, the data that I'm showing are based on the population of CFW mice, which are outbred stock mice, and we've measured weights of hind limb muscles in these animals. And a very large number of these animals we have examined. What emerged from this analysis is that, as in humans, variability in muscle mass is very substantial in this mouse model. So gastroc weight on the y-axis, in males it can range between 200 or over 200 to less than 100 milligrams. So again, it's two-fold difference in the weight. We also genotype these animals for a large number of SNPs, and that allowed us to do a genome-wide association analysis. And that is a very powerful method because it's an unbiased analysis. You can go and find genes that affect this variability in the actual population of healthy animals without even knowing that these genes exist. And the outcome of this analysis was quite fruitful in this case. So here we have a Manhattan plot of the on the weight of the gastrocnemius muscle, and what we can see that we have at least 3 regions in the genome on chromosome 6, 11, and also on chromosome X that are associated with the variability in muscle weight. So all of them are affecting muscle weight of the gastrocnemius in these animals. I'm going to focus on this particular region on chromosome 6 today because one of the questions that we would be asked when we find these associations is also, can you validate that the genes that are located there really affect the phenotype? And in this case, the only gene apparently that was present in this region of the chromosome was the MET gene. So it encodes a receptor for hepatocyte growth factor known as a c-MET. And we didn't have to produce a knockout model to show that the gene has something to do with skeletal muscle. These studies have been done a number of years ago, and they show that in the knockout model, there is no skeletal muscle developing in the limb and diaphragm. So apparently the gene is relevant for development of the skeletal muscle. We also found that there is a polymorphism that results in the substitution of amino acids. So arginine is substituted to cysteine in this population. And many animals would have either one or the other allele. And the SNP that was really responsible for this has one of the highest association value, or the lowest p-value with the muscle weight. So then we said we want to go and investigate, so what is really effect of this MET gene, how these effects are brought about, and what is happening at the level of the muscle fibers. So for this analysis, we took a soleus muscle as a model because it was also affected by the same gene as the gastrocnemius. And when we looked at the mean of all the animals that would be homozygous for the arginine allele, or the mean against the mean all homozygous for the cysteine allele, there was obvious that animals with the cysteine allele, they had a lower muscle weight in the soleus. So then we selected a subsample, which is shown here in white bars, of these soleae muscles. And we made sure that we chose from this pool soleae that were similar to the mean rather than the highest. And the other variable, so that was the same for both groups. And the other factor that we looked into and controlled, we made sure that these two other regions on chromosome 11 and on chromosome X, these animals would have the same allele. So the differences that we see here should be primarily attributed to this MET gene. Also, it appears that the effect is primarily muscle specific. It has not much to do with the growth in general, because the length of the bone, the tibia, where these muscles are attached to, they are pretty much identical between the two groups. So then we did histology. We sliced the muscle in half, cross-sectioned, did a TPA staining, acid pre-incubation, and then just counted type 1 and type 2 fibers, and also measured cross-sectional area in 20% of the fibers of each type. And the results of this analysis indicated that if we look at the number of fibers, and particularly number of type 2 fibers, so the carriers of the cysteine allele, they had about 20% lower number of type 2 fibers in the soleus muscle compared to the carriers of the arginine allele. There was no really difference in the number of fibers of type 1 fibers between these animals. Cross-sectional area of the fibers not affected by the allele. Doesn't really matter whether you have arginine or cysteine, very similar cross-sectional area. So we wanted to check if we can reproduce the same finding in females. We did so exactly the same protocol with the female samples of the soleae, and the same thing emerged again. So we had a reduction in the number of type 2 fibers in those animals that carried the cysteine allele. The reduction was a bit more profound, about 30% decline. Again, no difference in the number of type 1 fibers, nor there was any difference in the cross-sectional area of the fibers. So what we concluded out of it is obviously that allelic variant of the MET gene affects soleus weight via the influence on the number of type 2 fibers. So what are the implications of this analysis and these results? So we have seen this equation earlier in several presentations over this meeting, so that the phenotype is affected by genome, environment, and gene by environment interaction. So obviously what we have tried to do here helps us to tease out specific genes that are responsible for this G in this equation. And so it helps to sort of understand the underlying genetics of the phenotypes that are relevant for the variables that are important for athletic performance. So it's obviously what appears from this is that variants of MET can affect proportion of oxidative fibers in skeletal muscle. And that, and also the fact in itself that it would affect the total number of fibers, help us to understand, why we have individuals predisposed to some types of athletic activities compared to other type of athletic activities. So, and even though we haven't really analyzed the effects of this gene on exercise, it can help to understand how this can be relevant in predicting exercise performance. Finally, I want to acknowledge people that contributed to this work, students at the University of Aberdeen, and also my collaborators here in the UK and also in the United States, and support from the National Institute of Health. Thank you. I'm happy to take questions.